Hello, my name is Schwarz and I'm a PhD student in the Sea Lion project Galápagos at Bielefeld University. In this video abstract for current biology, I will showcase how we use Vibrissae length of Galápagos sea lions to study their adaptation to climate change. Our study takes place on the small island of Camaño, located in the center of the Galápagos archipelago close to the island of Santa Cruz. Here, we studied the behavior of Galápagos sea lion females in the ocean by equipping them with biologic devices. The collected data enable us to identify two major strategies: pelagic foragers that find their prey in the open water column and benthic foragers hunting for prey on the sea floor. After having a closer look, we found that benthic foragers swim mostly parallel to the sea floor, to then frequently go head first towards the substrate. As a lot of the benthic prey species identified through scatter analysis are fish species that hide in the sediment during the day, we hypothesize that benthic foragers are digging in sand to catch their prey. This hypothesis is strongly supported by the fact that benthic foragers had bald patches of fur on their nose and chin, likely due to abrasion as a result from their specialized foraging behavior. In addition, the Vibrissae length of benthic foragers was significantly shorter than the Vibrissae length of pelagic foragers. Here you see, for example, a typical benthic forager with short Vibrissae on the left and a pelagic forager with long Vibrissae on the right. To quantify these differences, we took pictures of adult females in profile and calculated the relative Vibrissae length by dividing the distance from the nose to the tip of the longest Vibrissae with the distance taken from the nose to the eye. Using this approach, we could now determine the foraging strategy of an adult female just based on the picture. But why is this observation so exciting? As we can now use Vibrissae length as a proxy for foraging strategies, we could demonstrate a stability of Vibrissae length and thus foraging strategies over longer time periods by taking repeated pictures of the same individuals. This also means we could significantly increase our sample size without the need to deploy further biologgers. As this colony has been part of a long-term monitoring project, and we have life history data of individuals since 2003, we can now combine the information of foraging strategies with the data of the reproductive success of females. We have therefore now the opportunity to compare the fitness consequences between benthic versus pelagic foraging adult females over the last years, and therefore also over different environmental conditions. Here you see the pupping probability and the body condition of offspring of benthic foragers in blue and pelagic foragers in red over different sea surface temperatures. As you can see, reproductive success of pelagic foragers is higher during colder temperatures, but decreases when temperatures rise, while benthic foragers' reproductive success is initially lower but stable with increasing temperatures. We thereby observed a fitness trade-off between the strategies, which is dependent on the environment. With two strategies being differently adapted to different environmental conditions, this foraging polymorphism can buffer the negative effect of environmental change in Galápagos sea lions. However, using the information of the strategy-dependent reproductive success in demographic models, we could demonstrate that this buffer effect is not enough to reverse the ongoing decline in our study population. We could also show that the increase in sea surface temperature is not explaining on its own the observed decrease in population size, and that we need to consider additional factors such as overfishing or other anthropogenic disturbances to understand why we have each year less animals. This information is essential if you want to optimize conservation efforts of this endangered marine mammal.